So lab reports are going to be a pretty important part of your kind of overall mark in Science 10. So you're going to need to really follow along here so that you know what my expectations are in terms of what you need to be able to produce as a proper lab report. Okay, because this goes in that 45% of your mark section that is the course work. Okay, that'll be labs and assignments and projects and stuff like that. But a lot of it is your lab reports. Okay, you're going to have between probably closer to 3 per unit. Okay, as long as we have time to do all 3 in each unit, but definitely 2, if not 3, per unit in this course. So that's, it adds up to a few lab reports, and it's something that we need to know how to do. And it's important because this format carries through into your 20 level sciences when you go there. All right. So the first thing is that when we do a lab, you will get in the stream of Google Classroom, I'll put in an assignment that says, properties of compounds lab, which is what one we're going to do next week. And in there will be a PDF file that'll be like the lab sheet with all the instructions and everything, and a link to the podcast where I explained it or whatever. And then there'll be another thing in there that is a Google Doc, and it actually has your name on it. It'll say your name, properties of compounds lab. That's where you're going to do your lab report. It's your file, okay? And I've already built it. Okay, so it already has the like it'll say problem, but it'll be blank. Okay, it'll say design, manipulated, responding, controlled. It'll be blank beside those. Okay, and basically you have to fill it in, but at least the format is already set up for you. All right, so you want to work in that file. In fact, you have to work within that file because I won't accept it any other way. Okay, if you go home and make up your whole own lab report on your Mac and you try and submit that to me, there's no way for me to open that. I don't have a Mac, so I can't open it, okay, and it's going to create problems, right? This way, I can track what was going on. I can see what you did. That helps me to find if you have any misunderstandings, because I can go back through your revision history and kind of see what you were doing, right? So that's the way it's going to have to be done, is through Google Classroom on Google Docs in this case. Okay, is everybody clear there? All right. Hopefully, having the format already set up is going to save you work, and that's the idea. Okay, so to start any experiment, we've got to start out with a problem that we need to test. Now, your sheet will have that either blank or it'll have it written there and you'll have to restate it. Either way, you need to restate it in the form of a question in your own words. So let's say that the problem that we're investigating, for example, is what is the effect of light on plant growth? Okay, well, you could restate that easily as how does light affect the growth of plants? Okay, or something like that, right? You just put it in your own words. That's the problem that we're investigating, right? The next thing that comes in are your variables, all right? So you're not only going to have to identify the variables, but you're also going to have to explain why you said this one's manipulated, that one's responding, or these ones are controlled, okay? How many manipulated variables are there in an experiment? One. There can only be one. If you test more than one thing, you'll never know which of the 2 things or 3 things you tested actually caused the change in the experiment. So the whole purpose of having a controlled experiment is to test the effect of only one thing. Okay, so in this case, I only want to test the effect of light on plant growth. All right. Now that doesn't mean I'm going to have such a simple experiment that it's this plant's in the dark and this plant's in the light and that's the end of the experiment. I can test varying amounts of light. I'm still only testing the effect of light. Okay, pharmaceutical companies, when they test new drugs, okay, here's what they do. They don't just say, all right, you people are getting a placebo, which is a sugar pill, and you guys are getting the real pill, and then we're going to compare at the end, right? They might say, okay, here's my group that doesn't get any of the drug, because I need to see what happens if you don't take it, i.e. you don't get better probably, okay, or whatever it happens to be, okay? And then these people are going to get this dosage. These people will get a stronger dosage. These people will get an even stronger dosage, and then you can kind of check, did the drug have any effect at all, and which dosage was the best? Okay, so you can test that, but you're still really only manipulating one thing. These people either got it or they didn't. Okay, so there's one manipulated variable. So what I want you guys to do in a lab report is, here's the manipulated variable, here's how it's being manipulated, and the effect. So I would say in this, so it's the part of the experiment that's changed on purpose by the experimenter, what's being tested. So as an example, you would write something like the amount of light given to plants because all subjects will receive different amounts of light and light powers photosynthesis. So I said, here's the variable, here's how I'm manipulating it, and the effect I think it'll have. All right, so those are the things that go in your explanation. If all you write down is light, I'm probably not going to be terribly impressed. And as a result, my marking hand will probably not go into the higher numbers. Okay, it will probably stay low. All right, I need more explanation than light. Okay, next thing is your responding variable or variables. Is it possible to have more than one responding variable? Yeah, absolutely. If I'm watching the growth of plants, I'm probably not just measuring one thing, right? If the only thing I measured in terms of the growth of plants was how tall they got, that's a pretty narrow view of plant growth overall. I would watch several things. I would measure not only how tall, I would count the number of leaves that it grew, and I would probably make some qualitative observations as well, like plants that were in the sun for a very long time tended to look kind of yellow or bleached, and plants that were in the dark a lot tended to look dead or whatever. But I would have like some qualitative observations that would also be part of my set of responding variables. So it is possible and actually quite common to have more than one responding variable. All right, so what we would write here is, okay, so what is the responding variable? Well, the responding variable is what changes as the result of the manipulations made by the experimenter. You wouldn't have to write that, that's just the definition. Okay, what you would write would be the growth of the plants. Okay, that's identifying here. This is my responding variable, the growth of the plants. Okay, because plants getting more light have more energy for growth. All right, so I've said, here's the responding variable, and here's how it's going to respond, and here's why. Okay, everybody follow? And like I said, in a real experiment, you would probably have a few more things. You'd be more specific about the growth of plants. You might say the growth of plants measured by height, number of leaves, color, whatever else you're measuring. Okay, controlled variables. The expectation on controlled variables is that you are able to identify three controlled variables for every experiment. Now, what's the importance of a controlled variable? Yeah, I got to keep it the same. Controlled variables are the things that could interfere with my lab if I didn't keep them the same for all of the subjects in my lab. All right, so as an example with this one that we're doing, okay, we would say, I wanted to use the same species of plant for all my subjects. It's hardly fair to compare the growth of a bean, a carrot, and a spruce tree. All right, that's not a fair experiment. I mean, they grow at different rates, and that doesn't make any sense. I would probably plant like five identical bean plants or something like that, so that I could have a fair comparison. Okay, so we would write that out as same species of plant because different plants grow at different rates. Okay, there's an explanation. It would affect the experiment in this way. Here's a controlled variable, and here's how it would affect the experiment if it wasn't controlled. Okay, and you might sometimes also write in there how you're controlling it. I mean, same species of plant. I'm not using different plants. That's kind of a given the way I wrote that. All right, another controlled variable for this experiment, the same amount of water. Water affects the growth of plants. Okay, if I water them all differently, I don't know then whether it was the light or the fact that some of them, shriveled up and died because I didn't water them. Okay, they all need to receive the same amount of water. Okay, same amount of water because water is an important factor in plant growth. So each plant needs the same amount, so it can't affect the growth rate of plants. Right, and you might even mention in there how much each plant is getting, 20 milliliters per day, okay, something like that. All right, another controlled variable for this experiment, same soil. All right, it's hardly fair to plant the different plants in, one in this potting soil I bought at the garden center, one in a pile of rocks, and another in a bunch of sand I dug out of my kid's sandbox. Okay, hardly, again, a controlled experiment. Now I don't know whether it was the dirt they were in or whether it was a different amount of light. So they all need to grow in the same soil. All right, so each pot has to have the same kind of soil in it. All right, and we would list that because plants get nutrients from soil and different soils hold water differently. Keeping them the same ensures that it cannot affect the experiment. All right, other things, I mean, I'm listing 4 here. I only expect you to list 3. Same temperature. Right, I want to have one plant that's in freezing temperatures and one plant that's in a tropical environment and things like that because, again, it's not a fair experiment. Okay, plants typically slow their growth in extreme heat or cold. Choosing an effective temperature for all plants ensures it cannot affect the experiment. So as I said before, identify the controlled variable, tell me how you're controlling it and how it would affect the experiment. Okay, that's what's expected for each of those. All right, this is the part that typically is the hardest part for people when they first get to Science 10 is how to make a proper hypothesis. Because probably up until now, you've been told that a hypothesis is a guess. It is so not a guess. I want to strangle whoever's teaching you that. Okay, it is not a guess. It is a statement, right? You have to put out there in your experiment that here's what I'm doing, and this will be the result of it. Okay, you have to put yourself out there. You have to set yourself up to be right or wrong, one or the other. You can't sit on the fence in science. Okay, you want to know, am I right or wrong at the end of this experiment, right? And it isn't a guess. You're going to have reasoning behind what you say. Okay, so to that end, and a hypothesis will always contain these three words: if, and, then. The if part is talking about the relationship you're investigating. All right, what's the relationship between the things you're investigating? All right, the and part describes the experiment briefly. You don't want to write out the whole experiment. That's what the procedure is, just a brief description of it. The then part is, here's the results I'll see if I'm right. Okay, so if this is true and I do this, then this is going to happen. That way you'll know I'm going to be right or wrong at the end of this experiment. Okay, it's a statement, it's not a guess. All right, does everybody follow the difference there? I know up until now you've been taught it's an educated guess, but it's so not. Okay, it's a statement of probability. All right. So as an example here, okay, what we would say is, if plants require light for growth, because that's what we're saying, how does light affect plant growth? They require it for growth. So if plants require light for growth and 5 identical plants are given different amounts of light, then the plants that receive the most light will show the most growth. That's actually a fairly simplistic hypothesis, but I think it gives you the general idea here. Agreed? Okay. Just looking at the way I've worded that, am I going to be accepting this hypothesis after the experiment or rejecting it? It's saying that the more light they get, the better they'll grow. If you put a plant in 24 hours of sunlight for a month on end, is it going to like that? Probably not. Okay, I'm probably not going to be accepting this hypothesis after I perform the experiment. Okay, does everyone follow how that works? Okay, so it's really important that you understand what each section of the hypothesis is about. Okay, the if is what's causing the problem, or what's the relationship being investigated. I'm saying the relationship is light makes plants grow. All right, that's what I'm saying. The and part briefly describes the experiment. I just said 5 plants are going to get different amounts of light. Okay? I would probably be more specific than that. I would say that, over the course of a month, 5 plants will receive different amounts of light, but the same amount of water or whatever. Okay, and the then part, what do you expect if you are correct? Well, I expect that the plants that get the most light will show the most growth. I've put it out there unequivocally. That's what I expect. I'm going to be wrong, and that's okay. Right? In fact, in real science, you're going to be wrong with a hypothesis probably 19 out of every 20 times. Okay? That's how science works. Doesn't mean you're totally wrong. You're going to learn something each time. Then you modify and you try again. You modify and you try again. Okay? That's how the process is supposed to work. In terms of marks, I'm mostly looking at format when I look at your hypothesis. I'm not going to dock you marks if I know your hypothesis is going to end up being rejected. Like I said, most are. Okay? If your hypothesis ends up being wrong, you're not going to be docked marks for that. Don't, after you're done the experiment, go, oh crap, my hypothesis is wrong. I better go back and change it. Don't. Okay? It's okay. If it was in the right format, I'm good with that. If it was if, and then, and you said what you were going to do, and this is what the results were going to be, that's what I'm looking for. Okay? All right, and generally we try to avoid the use of I, us, or we in a hypothesis. You'll notice that I didn't have those words in mind. Okay, if plants require light for growth and 5 identical plants are given different amounts of light, then the plants that receive the most light will show the most growth. I never mentioned me or anybody else in that thing, because if you do, then really for anyone to repeat your research, you have to be there, because you were part of the hypothesis and you don't want to be. Okay, so try and keep yourself and other people out of it. All right, for the procedure, sometimes you'll have to make up the procedure. right? The first lab we do, I'm going to show you the tests that you're going to do, and you're actually going to write up the procedure, but that's rare. Rarely do you actually have to write up the procedure. Most of the time, I've got it set up step by step by step. You follow the steps, okay? So in that case, you won't have to write anything for the procedure. Once in a while, you'll have to write the procedure yourself. After the procedure is your observations, okay? So you might have a chart where you've recorded your observations, or maybe you've got some pictures that you took of a plant while it was growing, or diagrams that you drew, or whatever. All of those count as observations. They were the data and other things that you collected while the experiment was running. There is a section of the lab report where that stuff goes. It's right under procedure, because it's the procedure that gets them for you, okay? After the observations is your analysis section. In a real lab, okay, you would be looking at your observations, looking at what you said in the hypothesis, and analyzing your observations. What do my observations say? Okay, so let's say I ran this experiment, and what I found was plants that got less than 6 hours of light died. Plants that got between 6 and 18 hours of light grew very well, with the ones at the 18-hour mark doing the best. And plants that got more than 18 hours of light died. Okay, let's say that those were my observations. Okay, that's not really realistic, but let's just say that's what they were. All right, so I'm looking at those observations and I'm analyzing those, and I can tell right away that more light is not necessarily better. It's better to a point. Okay, there, I've just analyzed my results. Okay, my results say that there's a point where there's too much. There's also a point where there's too little. All right, and that's something that I'll have to look at in terms of maybe a further experiment. Okay, for you guys, most of the time what analysis will be is, here's 4 or 5 questions. Using your observations, answer them, and it'll kind of be guiding you through that analytic process. Okay, again, the questions will already be on your template. You don't have to write them in or anything like that. You just have to answer them, okay, or do the calculations they ask you to do, things like that. Okay, after the analysis is your conclusion. The first thing you do in your conclusion is restate your hypothesis. In other words, copy it from the top and paste it back down below here. Save yourself some time. Okay, so copy and paste it down below. Then you need to provide me an explanation, and that explanation should be like a short paragraph. Okay, here is my hypothesis. This is what it said. My observations and analysis seem to say this. Therefore, my hypothesis is either acceptable or I reject my hypothesis on these grounds. All right, so you have to say, here's what I saw, here's what it means, and this is why I accept or this is why I reject my hypothesis. If you just write, I was right, well, goody for you, okay? But I was right is not a conclusion, okay? You have to tell me, how do you know you were right? Why do your observations say you were right? Nobody's ever 100% right in science, okay? What was correct? What worked? Okay, so does everyone understand the conclusion part? Okay, and the very last part is the evaluation. You need to identify 2 sources of error and suggest improvements. These sources of error are design errors. They are not performance errors. Okay, and so I'll explain the difference between the two. Okay, a design error would be something like this. I ask you to do an experiment where you're observing chemical reactions. You're mixing 2 chemicals together, and I give you like a contact lens sized dish to do the experiment in. Okay, how hard is it going to be to properly observe a chemical reaction in a sample size that small? Pretty difficult. Okay, is that a source of error that was beyond your control? Yeah, it was. I told you this is what you use and I gave it to you. Okay, it was the design of the experiment. It interfered with your ability to do the experiment correctly. So you can list in there, source of error. Size of the sample was really small. This made observations of the reactions difficult. A larger sample size would make observations easier. Okay, it's not going to hurt my feelings. We designed the labs to have sources of error for you to find. Okay, what I don't want to see are performance errors like the following. When I did trial 2, I spilled some of the other stuff in it, and so that affected my observations. Okay, here's what that sounds like in teacher speak. I screwed up and I was too lazy to do it over. Thus, my whole experiment is now invalid. We don't want to send that message to me when I'm marking, because then I'm going to go back and go, oh, really? well, that one's no good and that one's no good. Okay, so yeah, we don't want to be saying, hey, Mr. Cordero, I screwed up and I didn't fix it. Okay, if you screw something up during the experiment, and that happens, go back and do it again. Okay, you have to in order to get good data. You can't write in there that you screwed up. Okay, you have to make sure you've done it correctly. Does everyone follow the difference between a design and a performance error? Okay. All right, so you need 2 of those and ways to improve them. So when you first start out doing these reports, the first couple are probably going to take you a little longer to do. Okay, I would say that when you go home and you're putting them together, you're looking at a half an hour to 40 minutes probably of work in order to get them done for the first few. Once you get the hang of the format, you'll go a lot quicker. All right, and you're probably by the end of the semester, or middle of the semester, probably be doing a lab report in between 20 minutes to half an hour tops. Okay, because you get some class time to work on it. Right? If we go over and we're doing the lab and we're done the lab, you come back over here and you work on the lab report a little bit. Okay, you can always come in and schedule help for lunch if you need to. Okay, but expect that lab report is going to be a bit of homework for you each time. But obviously it's not being made to be due the next day, right? I always give you a minimum of 2 school days for anything. And usually with lab reports, it's like 4 days, okay, something like that. All right, questions on the expectations for a lab report? All right, so it's got to be detailed. If you've got 1 word or like 3-word answers for things in a lab report, you're not being thorough enough, okay? You need to be more thorough than that. Okay, that might be acceptable in lower grades, but here we're looking for detail. Okay, we're looking for you to see the details that are in there and report them back. Question? Well, for sure in the hypothesis, we want to avoid doing that because it involves us in any further experimentation. So we want to try to avoid that. If it's in the observations, in the analysis, I mean, you might use, did the following things as part, that would be okay, right? But in the hypothesis, we try to avoid it. Yeah. Okay, any other questions there? Good. So tomorrow, I'm going to teach you about properties of compounds. That's going to be the stuff you're going to need in order to actually do the analysis of the data we get in the lab on Tuesday. All right, so tomorrow I'll teach you that stuff. Monday we'll sit down and we'll get started on the lab report. Okay, we'll work together on this first one so you kind of get the hang of it. You and your group will design your procedure and get like a chart for observations and stuff like that ready for the lab on Tuesday. Tuesday we'll perform the lab. It should go fairly quick, and hopefully we'll get back here and you can work on the report a little bit on that day as well. Okay, but that'll be the only class time that you'll get to work on it. Okay, so there's still a couple of minutes left, about 5 minutes left. So if you're not done that naming worksheet, make sure you finish that up. Okay, there's more naming worksheets in that workbook. If you're struggling with that or you want a little extra practice, that's what they're there for. Okay.